We'll now begin with the third and the last part of the series of inflammation lectures, wherein we'll discuss what happens if our body has to face the enemy or the foreign bodies for a longer period than required, and how does our body cope up with the aftermath of this kind of a war. We will discuss about chronic inflammation and tissue repair. As always, comments, questions and healthy discussions are always welcome in the comment section below, because knowledge shared is wisdom gained. So let's begin. Let's begin by understanding what's the definition of chronic inflammation. Chronic inflammation is defined as the response of a prolonged duration in weeks or months in which inflammation, tissue injury and attempts at repair coexist in varying conditions. So it's very important to understand that acute inflammation, as discussed in the previous videos, lasts for days to weeks. But if the response is for a prolonged duration, that is weeks or months, it comes under the category of chronic inflammation. What are the causes of chronic inflammation? Persistent infections, as in case of tuberculosis, hypersensitivity reactions, which will lead to conditions like rheumatoid arthritis, or exposure to any toxic agents for a prolonged period of time, can result in chronic inflammation. What are the classic morphological features which you will encounter as soon as you see a slide showing chronic inflammation? There are three classic features. One is infiltration by mononuclear cells. By mononuclear cells, I mean lymphocytes and macrophages. There will also be tissue destruction and there will be attempts at healing. Attempts at healing include formation of granulation tissue, healing by primary or secondary intention. What is the meaning of all this? We will elaborate in the further slides. For starters, let us try to understand the role of macrophages in chronic inflammation. Macrophages are the dominant cells in chronic inflammation, which contribute by secreting cytokines and growth factors, destroying foreign body and activating the T lymphocyte. All these functions will be discussed in detail in the forthcoming slides, but before that it is very important to understand how they undergo maturation and come into action. They have two sources of development. One is the hematopoietic stem cell from the bone marrow in the postnatal life, and the second source of development is the yolk sac or the fetal liver in the prenatal life. Now all these will result in formation of activated macrophages. But there are two ways of development. One is, like I said, from the bone marrow. They arise from something called as the blood monocyte. That is monocytes which are released from the bone marrow and hematopoietic stem cell reach the blood vessel and they form the activated macrophages. Now these activated macrophages are always present in sites of inflammation in skin and in intestinal tract. Now the ones that arise from the yolk sac or the fetal liver, they reside in a particular tissue at a very early life, and these are named particular macrophages depending on the location. Like for example, the Kupffer cells are nothing but macrophages of the liver. Alveolar macrophages are nothing but the macrophages present in the respiratory system. Microglia are macrophages in the CNS, etc. Now that we know how these macrophages develop and mature, let us look at the pathways of macrophage activation. So there are two pathways. The first is a classically activated macrophage or the M1 pathway, which is induced, as you can see here, by microbes, toll-like receptors or IFN gamma, which generate two products. The first is reactive oxygen species, nitrogen oxygens or lysosomal enzyme, which engage in microbicidal actions, which is phagocytosis and killing of many bacteria and fungi. They also generate cytokines like IL-1, IL-12 and chemokines, which help in inflammation. The alternatively activated macrophage pathway or the M2 pathway is induced by IL-13 and IL-4, which is from the T lymphocytes. Now what they do is they will generate growth factors and TGF beta, which help in tissue repair and fibrosis, and they also generate IL-10 and TGF beta, which have anti-inflammatory effects. It is very important to activate T and B lymphocytes to initiate and propagate inflammation. Now, how does this macrophage and T lymphocyte interact with each other? Before understanding that, it is important to understand what are CD4 plus Th1 cells. CD4 plus T cell are a subset of T cells that promote inflammation due to their ability to secrete cytokines. There are three subsets of cytokine that CD4 plus secretes. The first is the Th1 cell, which will activate the M1 pathway. The second is the Th2 cell, which will activate the M2 pathway. And the third is the Th17 cell, which will secrete IL-17, which is responsible for chemokines. Now, macrophages interact with the CD4 plus T cell by presenting the antigen to the CD4 plus T cell. That's why they are called as the antigen presenting cell. Apart from presenting the antigen, they also present molecules called co-stimulators. Now, what are co-stimulators? That we will explain in a topic called as autoimmunity. Now, once that is expressed, this CD4 plus T cell will express the IL-12, as you can see here, that will stimulate the T cell response. How does it stimulate T cell response? By recruiting more activated macrophages, and that will lead to the sustained chronic inflammation. The sustained inflammation that was mentioned in the previous slide is nothing but granulomatous inflammation. What is a granulomatous inflammation? It's a chronic inflammation which is characterized by collection of activated macrophages with T lymphocyte and sometimes associated with necrosis. There are many types of granulomas that can be formed. The first is the foreign body granulomas, which is due to inert foreign bodies like talc or sutures. The second is immune granulomas, which will occur due to a persistent T cell response as seen in schistosomiasis. The third are TB granulomas, which is as self-explanatory as it is found in TB, and they are caseating granulomas. Now that we've seen what happens when there is sustained inflammation in the body, let's now see how our body combats this inflammation. Our body combats the inflammation process with the help of a process called as tissue repair. Tissue repair is nothing but restoration of tissue architecture and function after an injury. Tissue repair has two major mechanisms. The first is regeneration. Regeneration usually occurs wherein the tissues are able to replace the damaged component and return to its normal state, as you can see in this photo. The second is scar formation. Scar formation occurs if it's incapable to regenerate. So what it does is it will lay down layers of connective tissue, and organization will also happen if there is inflammatory exudate present. For tissue repair to occur, the core is always cell proliferation. Cell proliferation has three different mechanisms depending upon the tissue. Now, what is cell proliferation? Cell proliferation always depends upon the growth factors and the integrity of the extracellular membrane. Now, the three mechanisms are, one is when it's a labile tissue. What do you mean by labile tissue? When the tissue has continuously dividing cells which are lost and replaced. Like for example, the hematopoietic cells in bone marrow and the majority of the surface epithelia. The next is when it's a stable tissue. What do you mean by stable tissue? When they have minimal proliferative capacity. For example, in solid tissues like liver, kidney and pancreas, endothelial cells, fibroblasts and smooth muscle cells. The third is in when they are permanent tissues. Permanent tissues will have terminally differentiated and non-proliferative cell. For example, majority of the neurons and cardiac muscles are terminally differentiated and non-proliferative. One of the best ways to understand the concept of cellular proliferation is to understand the concept of liver regeneration. So what happens is whenever the liver undergoes an injury, there will be hepatocyte proliferation which is dependent upon cytokine products and polypeptide growth factors. There are two phases to liver regeneration. The first is this, which is called as the priming phase, wherein you have the Kupffer cells which usually secrete the IL-6 and they will respond to growth factor signals. The second phase is this growth factor phase, wherein you will have HGF as well as the TGF alpha, as you can see here, which will act upon these primed hepatocytes, and this will lead to the entry of the cells into the cell cycle, and that would lead to cellular proliferation. Once we've understood the concept of cellular proliferation, let us now see how connective tissue is deposited and how a scar is formed. So as you can see here, you will have activated platelets. These platelets will secrete chemokines, and these chemokines will recruit neutrophils and monocytes for the next 6 to 48 hours, which will eliminate the microbes. Cellular proliferation will occur for the next 10 days, wherein you will have epithelial cells which will usually respond to the growth factors. The endothelial cells will engage in a process called as angiogenesis. What is angiogenesis? It is formation of new blood vessel, a concept that we will explore in the further slides. You will also have fibroblasts which will lay down the collagen fibers, as you can see here. All these will form the granulation tissue, which is proliferation of fibroblasts, the new capillaries as well as the macrophages. As discussed in the previous slide, angiogenesis is nothing but new blood vessel development from the existing blood vessels. There are various steps and mechanisms to angiogenesis, but before that, let us understand the structure of a quiescent vessel. Now, in a quiescent or a resting blood vessel, the inner layer is the endothelium, the outer layer is a pericyte, and the one separating them is the basement membrane. Due to various angiogenic factors, which is NO and increased VEGF, there will be vasodilation. Now, vasodilation will then lead to separation of pericytes. Then there will be migration and proliferation of the endothelial cells. There will also be remodeling into tubes, recruitment of the periendothelial cells, and suppression of the endothelial proliferation. What is a mechanism? So the mechanism, the first molecule like we discussed before is VEGF-A. Now VEGF-A will cause migration and proliferation of the endothelial cells and vasodilation with the help of nitric oxide. There is a molecule called FGF2, fibroblast growth factor 2, FGF2, which will act upon these endothelial cells. They will cause migration of macrophages as well as fibroblasts. There is angiopoietin 1 and 2, which will cause maturation of the blood vessels. There will be PDGF, which will cause smooth muscle cell recruitment, and TGF beta, which will suppress the endothelial cells. Now, this PDGF and TGF beta is activated by the Notch pathway. Notch pathway will cause sprouting of the new vessels. After angiogenesis has taken place, the next role is the deposition of connective tissue. There are two mechanisms to this. One is migration and proliferation of fibroblasts, and the second is deposition of ECM proteins. This is orchestrated by TGF beta. So what are the functions of TGF beta? To summarize, like we saw before, it basically stimulates fibroblast proliferation. It will inhibit the degradation of ECM. The third is it will synthesize collagen and fibronectin. The fourth is it will cause anti-inflammatory properties of cytokine, that is it will decrease the inflammatory response. After connective tissue deposition, the next step is remodeling of the connective tissue. Remodeling of the connective tissue is brought about by molecules called as MMPs. MMPs are nothing but matrix metalloproteinases. So, what do these do? There are various forms of matrix metalloproteinases, of which we have to know what is MMP 1, 2 and 3. Now, MMP 1, 2 and 3 is very essential because it helps in cleaving of the collagen. How are these MMPs activated? Basically, when you have inactivated zymogen, which is activated by plasmin, it leads to formation of MMPs or matrix metalloproteinases. Now that we've seen how tissue repair and fibrosis takes place, let us look at two of the most important examples of the same. The first is healing by primary intention and the second is healing by secondary intention. Let us explore their steps one by one. So for healing by primary intention, the first step is formation of a clot. Clot usually forms because of activation of the coagulation pathway. In the first 24 hours, as you can see here, these neutrophils will migrate to the site of injury and there will be formation of the epithelial cells which will close the wound. After 3 to 7 days, these neutrophils are replaced by macrophages and there will be formation of a granulation tissue. In the 5th day, there will be formation of leaky blood vessels. After weeks, collagen will get accumulated and there will also be fibroblast proliferation. Following this, there will be formation of a scar. Now, for healing by secondary intention, it is the same process as primary intention, but the scale is bigger. For example, there will be more exudate and necrotic debris, there will be a larger amount of granulation tissue, and there will also be formation from type 3 collagen to type 1 collagen. And because the scar is bigger, there will be scar contraction. So that means wound contraction will occur in healing of secondary intention. So this was all about inflammation. We have discussed acute inflammation, the mediators of acute inflammation, chronic inflammation, and tissue repair. Thank you for watching, and thank you for all the likes, the comments, as well as the views. Don't forget to like, share, and subscribe to my YouTube channel, and let's always keep up the process of sharing knowledge and gaining wisdom. Do let me know what other topics I should cover next in my YouTube channel, and see you all in the next video.